Understanding Neuronal Impulse Generation and Transmission. A comprehensive guide introduction. The human brain is a complex organ composed of billions of nerve cells called neurons. These neurons communicate with each other through electrical impulses, allowing us to think, move, and perceive the world around us. In this blog post, we will delve deep into the process of how neurons generate impulses and transmit them to other cells. Neuronal structure. Neurons have a unique structure that enables them to transmit signals effectively. They consist of three main parts: the cell body, soma, dendrites, and axon. The cell body contains the nucleus and other organelles necessary for the neuron's functions. Dendrites are branched extensions that receive signals from other neurons. The axon is a long, slender projection that transmits signals away from the cell body. Generation of action potential. The primary mechanism by which neurons generate impulses is through an action potential. This process involves changes in the neuron's membrane potential, which is the difference in electrical charge between the inside and outside of the cell. Resting membrane potential. When a neuron is at rest, its membrane potential is negative, typically around -70 millivolts. This resting state is maintained by the activity of ion channels, particularly sodium, Na+, and potassium, K+, channels. Depolarization. When a stimulus, such as a neurotransmitter binding to receptors on the dendrites, triggers the neuron, ion channels open, allowing Na+ ions to flow into the cell. This influx of positive ions depolarizes the membrane, making it less negative. Threshold potential. If the depolarization reaches a certain threshold, usually around -55 millivolts, it triggers the opening of voltage-gated sodium channels along the axon. This initiates an action potential. Action potential. The sudden influx of Na+ ions through voltage-gated channels causes a rapid change in membrane potential, making it positive inside the cell. This spike in electrical activity is the action potential. Propagation of action potential. Once an action potential is initiated at the axon hillock, it propagates along the axon toward the synaptic terminals. This propagation occurs in a series of steps, all or none principle. The action potential follows the all or none principle, meaning it either occurs fully or not at all. If the threshold is reached, the action potential is generated, otherwise, no signal is transmitted. Sodium influx and potassium efflux. As the action potential travels down the axon, voltage-gated sodium channels open sequentially, allowing Na+ ions to enter and depolarize each segment. Meanwhile, potassium channels open to repolarize the membrane by allowing K+ ions to exit the cell. Propagation speed. The speed of action potential propagation varies depending on factors like axon diameter and myelination. Larger diameter axons and myelinated axons conduct impulses faster due to reduced ion leakage and saltatory conduction, jumping between nodes of Renvier. Synaptic transmission. At the synaptic terminals, the action potential triggers the release of neurotransmitters into the synaptic cleft, the tiny gap between neurons. This process of synaptic transmission involves several key steps. Neurotransmitter release. When the action potential reaches the synaptic terminals, it causes voltage-gated calcium channels to open. Calcium ions, Ca2+, enter the terminal, leading to the fusion of synaptic vesicles containing neurotransmitters with the cell membrane. Neurotransmitter binding. The neurotransmitters, such as dopamine, serotonin, or acetylcholine, diffuse across the synaptic cleft and bind to receptors on the postsynaptic neurons, dendrites, or cell body. Postsynaptic potential. Depending on the type of neurotransmitter and receptor, the postsynaptic neuron may experience excitatory postsynaptic potentials, EPSPs, or inhibitory postsynaptic potentials, IPSPs. EPSPs depolarize the neuron, while IPSPs hyperpolarize it, making it less likely to generate an action potential. Integration of signals. The postsynaptic neuron integrates EPSPs and IPSPs from multiple synapses, determining whether an action potential will be generated at its axon hillock. This process, known as synaptic integration, plays a crucial role in information processing in the brain. Conclusion. The generation and transmission of neuronal impulses are fundamental processes that underlie brain function and communication within the nervous system. Understanding how neurons generate action potentials, propagate them along axons, and transmit signals at synapses provides insights into the complexities of neural networks and their role in cognition, behavior, and physiological regulation.